Hello students, biomolecule is one of the important chapter in 11th biology, and we are going to solve some of the important points for the coming NEET exam from this chapter. So students, let us start with nitrogen bases. So you know nitrogen bases are of two types, purines and pyrimidines. So two purines we have, adenine and guanine, and three pyrimidines we have, cytosine, thymine and uracil. So you must remember here that purines are double ringed always. Two rings will be there, double ring in purines, whereas pyrimidines will be of single ring. And also remember, purines 9 membered and pyrimidines are 6 membered. And nucleic acids basically are of two types, DNA and RNA, and both the nucleic acids are the polymers of nucleotides. Now what are nucleotides? Nucleotides are made up of three components. One is nitrogen base, any of these five nitrogen bases, pentose sugar, 5 carbon sugar, and phosphoric acid. When these three components combine together, it is going to create nucleoside. And you must also remember, when nitrogen base and pentose sugar are going to link, it is going to create nucleoside. And nucleic acid discovered by Mixture, and he named them as nuclein, and later on Altman given the name nucleic acid. And remember, DNA has only 4 nitrogen bases. Out of these 5, adenine, guanine, cytosine and thymine. RNA, adenine, guanine, cytosine and uracil. So thymine present only in DNA, uracil present only in RNA, and pentose sugar present in DNA is deoxyribose, and in RNA it is ribose. According to Chargaff rule, the number of purines in the DNA is equal to the number of pyrimidines. That's why the ratio of adenine plus thymine divided by guanine plus cytosine is equal to 1, because when the base pairing occurs, adenine always pairs with thymine with 2 hydrogen bond, and guanine always pairs with cytosine with 3 hydrogen bonds. And remember, phosphodiester bond is going to link between the two nucleotides. Two nucleotides are going to join by phosphodiester bond. So these are the three types of DNA, B, A and Z, and the length of one helix in BDNA is 34 Armstrong, ADNA 25, and ZDNA 46 Armstrong. The ZDNA, in the ZDNA, the sugar moieties are opposite, so diester bond will be in zigzag form in ZDNA. And to measure the DNA synthesis, we can use radio labeled thymine, and to measure the RNA synthesis, we can use radio labeled uracil. Remember that even ATP is one nucleotide. And according to Kim, while he was working on phenylalanine, he came to know that tRNA looks like L shape, and tRNA also looks like cloverleaf model. And DNase is one enzyme belongs to class called nuclease, which is going to break the DNA by hydrolyzing phosphodiester bond present between the nucleotides. So DNA breaks phosphodiester bond. And according to Watson and Crick, DNA is made up of 2 polynucleotide strands. Both are anti-parallel. One runs in 5' to 3', another runs in 3' to 5' direction. And between the polynucleotide strands, we can find hydrogen bonds between the two polynucleotide strands. And uracil, as uracil is a pyrimidine present only in RNA, so uracil can form nucleoside only with ribose but not with deoxyribose. So there is one nucleic acid act as a enzyme called a ribozyme. It is a single stranded rRNA. It is act as a catalyst by splicing RNA during protein synthesis. And remember about enzyme, holoenzyme is a combination of coenzyme and enzyme. What is coenzyme? Coenzyme means organic or metallo-organic molecules that are required for the activity of enzyme. And apoenzyme means the protein part of the enzyme is called as apoenzyme. Now, presence of enzyme in any biochemical reaction is going to lower the activation energy. So lower enzymes lowers the activation energy. And remember, protein will be having 3-dimensional structure, and there are certain bonds which are going to stabilize this protein structure. That includes hydrogen bond, ionic bond, van der Waals forces, covalent bond, and hydrophobic bonds. But remember, ester bond has least involvement in maintaining the 3-dimensional structure. Ester bond has least involvement in stabilizing the 3-dimensional structure of protein. Remember, when enzyme and substrate reacts, they are going to form enzyme substrate complex, which is transient and unstable. This enzyme substrate complex is unstable, so it will convert into product and resynthesize enzyme back. So some of the vitamins act as coenzyme. For example, vitamin niacin, in that vitamin niacin, NAD and NADPH are going to act as a coenzyme that increase the activity of enzyme. And remember, collagen is the most abundant protein in animal kingdom, and rubisco is the most abundant protein in plant kingdom. Very important. And enzyme activity decreases above and below optimum temperature. At optimum temperature, enzyme activity will be maximum, but more than this temperature or less than this optimum temperature, enzyme activity is going to reduce. Remember. And detergent is the largest industrial applications of enzyme in industries. Detergent manufacturing uses most of the enzyme. And remember, there is a step in TCA cycle called succinate converts to fumarate by enzyme succinate dehydrogenase. But we have one structural analog to succinate called malonate. Malonate is a structural analog, and that is going to act as a competitive inhibitor of succinate. Means malonate compete with the succinate to react with this succinate dehydrogenase enzyme. But when succinate dehydrogenase enzyme react with malonate, it is not going to form fumarate, and TCA cycle is going to stop. And remember, our lymphocytes, our WBCs are going to produce some glycoproteins. They are going to act as immunoglobulins or antibody, important part of our immune system. And isozymes and or isoenzymes are the enzymes that are going to catalyze the same reaction, but they are differ from each other. And remember, this is the structure, normal structure of amino acid, general structure, where alpha carbon atom having amino group and carboxylic group. Alpha carbon atom has amino group, basic amino group, and acidic carboxylic group. And you can find one R group. If this R group having is hydrogen, then that simplest amino acid will be considered as glycine. If the R group is a simple hydrogen, then that amino acid is glycine. And in arthropods and molluscans, the exoskeleton is made up of chitin, and that chitin chemically made up of N-acetyl glucosamine. N-acetyl glucosamine is the derivative of glucose. So this is the general formula of carbohydrate, CxH2O Y times, and we know in carbohydrate monosaccharides are going to form polysaccharides, and between monosaccharides, two monosaccharides, there is a bond called glycosidic bond. Lipase enzyme used in detergents, pectinase in fruit juice, amylase in textile industry. Remember this. And carbon is the most abundant element in plants. Carbon is the most abundant element in the plants, and in the dry weight of element we can find these four elements. That's why carbon, nitrogen and hydrogen are going to act as a framework elements. Carbon, nitrogen, oxygen and hydrogen. Sugars are classified into two types, reducing and non-reducing. Reducing sugars, basically they have free aldehyde group or free keto groups. These are the example: glucose, fructose, galactose and mannose. Whereas non-reducing sugar does not have free aldehyde or keto group. Example: sucrose, starch, gluconic acid, beta methyl glucoside. These are the examples. Now in lysosomes are the suicide bags of the cell. You can find many enzymes like hydrolase, protease, nuclease, glycosidase and lipase. Remember, these are the different enzymes are going to present in lysosomes. In that, hydrolase is going to maintain pH of 5, acidic pH. And remember, cellulose is a fibrous protein present in the plant cell wall, responsible for the structure, and it is the most abundant organic substance on the earth. Cellulose is the most abundant organic substance. Most abundant protein in animal kingdom is collagen. In plant is rubisco. This cellulose is the most abundant organic substance. In plant cell, middle lamella is made up of calcium pectate and magnesium pectate. Remember, inulin is a polymer of fructose, and it is present in roots and tubers of Compositae family to stores the food. Inulin stores the food. It is a polymer of fructose. We can find it in maximum in family Compositae. Pectin is a mucopolysaccharide present in plant cell wall. Lactose is a disaccharide made up of glucose and galactose. Maltose is a disaccharide made up of two monosaccharides, that is glucose. Sucrose is also a disaccharide made up of glucose and fructose. So maltose is homodisaccharide, lactose and sucrose is heterodisaccharide. And among the biomolecule, lipid is not a polymer. And the basic units of lipid include 3 fatty acids and 1 glycerol. 3 fatty acid and one glycerol. Whereas in phosphoglyceride, fatty acid may be saturated or unsaturated, that is esterified with glycerol. In addition to that, it is having phosphate group. And remember, chitin present in exoskeleton of arthropods. It is made up of N-acetyl glucosamine, that is N nitrogen containing polysaccharide. It is nitrogen containing polysaccharide. Our cell membrane has three types of lipids, that is phospholipid, glycolipid and sterol. Sterol nothing but cholesterol.